Just look at the whiteboard and try to understand the Frenster model. This Frenster model was proposed in 1965 to explain the mechanism of regulation of gene expression in the eukaryote. So look here, this structure, the DNA structure is the DNA double helical structure has been shown in the red ink here, and this histone protein which remains associated with this histone protein has been shown in this diagram in green ink. So this is the histone protein, a part of the DNA which has been shown here is complexed with the histone protein. This the DNA double helical structure is remaining is associated with this histone protein. So this histone protein, suppose that this part of the DNA is to be transcribed. What will happen? This histone protein should get removed from that part of the DNA which is to be transcribed. So according to Frenster, the DNA, the histone protein act as the non-specific repressor of the transcription. So the part of the DNA, suppose that this middle part of this DNA is to be transcribed, what will happen? The histone protein will get removed. This part of the DNA has become free of the histone protein. The histone protein has got replaced here, and the DNA has been exposed. The part of the DNA which is to be transcribed has been exposed, and it has been made free of this histone protein. So this part of the DNA becomes a double helical part of the DNA becomes free due to the removal of the histone protein from its structure. And then the question arises: what are the factors, and why does this histone protein gets free or get dissociated from the DNA? So is there any chemical or something else which is helpful in replacing the histone protein from the part of the DNA which is to be transcribed? Yes, there are some chemicals which are known as polyions. There are some chemicals which present in the cell, present in the nucleus, and these polyions play a very important role. Polyions are the chemicals which are required for replacing the histone protein from that part of the DNA which is to be transcribed. These polyions replaces the histone protein from the part of the DNA which is to be transcribed. And this polyion not only removes this histone protein, but it also makes a complex with this removed part of the histone protein, so that it cannot again get associated with the part of the DNA which when the transcription is going on. So these polyions are very important. These polyions are very important because they bring about the separation of the histone protein from the part of the DNA which is to be transcribed. And not only this, these polyions also make complex with this histone protein, so that this histone protein does not again this histone protein cannot again combine with the DNA and blocks the process of transcription. So this the part of the DNA which is to be transcribed has been exposed due to the removal or replacement of the histone protein by the help of these polyions. And now this part of the DNA which has been exposed gets open up. The hydrogen bond between the two helical structure breaks down, and these two strands of the DNA becomes free, and loops are formed, and the part of the DNA is transcribed. I have shown here the mRNA, which is hnRNA, which is being transcribed from this part of the DNA, and this is in the black ink. This mRNA has been shown in the black ink. So then what happens with the second R? The two strands are not transcribed at the same time. Rather, one of the two strands of the opened DNA helix acts as a template from which the mRNA is transcribed. What is the function of the other strand? This other strand, the part of the DNA from which the histone protein has been removed, one helix of the DNA, one separated helix, one of the separated strand of the DNA gets transcribed to form the hnRNA, which will be then converted into mRNA. And the other opened strand of this DNA is stabilized by another molecule which is called the D-repressor RNA. The D-repressor RNA combines with this single-stranded single strand of the DNA which is not being transcribed, and the another strand which is transcribed. So the strand of the DNA which is not transcribed is stabilized by a particular molecule, and that molecule is known as the de-repressor RNA. This de-repressor RNA stabilizes the another strand of the DNA which is not being transcribed, and to this in this way the Frenster's opinion was that it is the histone protein which acts as the repressor of the transcription and which prevents the transcription of the DNA. And for the transcription to take place, the part of the DNA which is to be transcribed should get dissociated from the histone protein. The histone protein should get removed or displaced from the part of the DNA which is to be transcribed. And this, what does it show? It shows that the histone protein also acts as the suppressor of the gene expression. And until and unless the histone proteins are not removed from the part of the DNA which is to be transcribed, DNA transcription cannot take place. So this is very important. This is a very important hypothesis which was proposed by the Frenster model. And according to this model, the histone protein acts as a repressor of the gene expression, and the gene which is to be transcribed, and the gene which means the part of the DNA which is to be transcribed, at first will need the replacement of the histone protein from that part of the DNA, so that the DNA can get exposed for the process of transcription. And when the DNA gets exposed, and in this process, the dissociation of this histone protein is brought about by the polyions. These polyions are very important. These polyions removes or displaces the histone protein from the part of the DNA which is to be transcribed. And the dissociated histone protein is stabilized, or is stabilized DNA. The dissociated histone proteins undergoes stabilization by the polyions also. So in this way, polyions play two roles. Polyions are very important because they help in the dissociation of this histone protein, and they make complex with the dissociated histone protein. And one of the two strands of the DNA, opened of the DNA, exposed DNA, is transcribed, and the another strand is stabilized by the presence of another molecule which is called de-repressor RNA. So this is the very simple explanation proposed by the Frenster with regard to the mechanism of the regulation of gene expression in the eukaryote. Let me know, am I clear to all of you?